I'm a PhD student at the Rosa Rademakers lab. I think Rosa is also here today, so she may later also join in on some of the discussions later on. And our lab is focused on frontotemporal dementia, and we look for causal genes and modifiers, and then we perform functional follow-up of these genes to try to figure out what they do, and then we also try to translate this to potential therapies for patients. And during my PhD, I am working on the biology and function of TMEM106B, which was identified as a risk factor and disease modifier for FTD back in 2010. Which means that I mainly focused on the two parts of functional follow-up and the translational research in the lab. And then before I dive into genetic modifiers and TMEM106B, I want to make sure that everyone can really follow along and that we're all on the same page. So I just want to go over a few introductory slides in which I wanted to explain some concepts or terms that I will later also frequently use and which will be important to understand the presentation. So as you know, our body consists of billions of cells and each have their own specialized function. And in these cells, we have, again, different compartments and all, yeah, I can't really show anything on the slides, but I will go slow enough so you can take a good look at them. So in the cells, we have, again, these different compartments, and all the cells have a center that we call the nucleus. And this contains our genetic code, or our DNA. And it's the genes that are read from this DNA that are transformed into RNA and translated into proteins. And if you will, you can compare a cell to a little factory, as in the DNA is sort of the management. So they carry all the information on what things need to be done and how they should be done. And this message is then distributed to, let's say, human resources, which is the RNA, and then they hire certain employees to carry out the job, and these employees are the proteins that carry out the job. And this genetic code is as the basis of everything and is passed on from generation to generation, and each gene has two copies. So you receive one from the father and one from the mother. But as you can imagine, a mistake in this genetic code, which we call mutation, causes the processes to be disturbed, and this will interfere with normal functioning of a cell, which is then harmful and may cause disease. So very briefly, our brain consists of different cell types with special functions. So we have neuronal cells and we have glial cells, and glial cells provide support to the neurons and protect them. And in diseases like frontotemporal dementia and, for example, also Alzheimer's, which we all call neurodegenerative diseases, it is, as the name already suggests, these neurons in the brain that are dying that cause the problems. So if you proceed to the next slide. So you may have heard of frontotemporal dementia, but also of frontotemporal lobar degeneration. And while both terms are referring to the same disease, frontotemporal dementia is actually used to describe the disease and the disease symptoms, while frontotemporal lobar degeneration describes what is happening in the brain. So in frontotemporal dementia, the neurons in the frontal and temporal lobes are degenerating or dying because of toxic accumulation and aggregations of proteins. And it is these aggregations that are very toxic and to which the neurons are very vulnerable. And the reason why neurons are very vulnerable for accumulation of proteins is because of their long lifespan and the fact that they do not regenerate. So this means that over time, as we get older, more and more proteins are accumulating, and once this reaches a certain threshold, this becomes toxic for the neurons and they die. This is causing problems as neurons are not dividing and therefore also not replaced when they are dying. This is in contrast to many other cells in our body, which have a short lifespan. They keep dividing our entire life, and the dividing helps diluting the accumulating proteins, but at the same time, they also replace the cells, so it's not a problem if some cells die. So actually, almost all neurodegenerative diseases is characterized by the accumulation and aggregation of these toxic proteins, and they differ in the type of protein that is forming the aggregates or in which region of the brains the neurons are dying. And also within one disease like FTD, you can have different subtypes based on what proteins are accumulating inside the brain. And for FTD, we have identified up to 3 different proteins that can accumulate, which is why we subclassify FTD as FTLD-TDP, in which a protein called TDP43 accumulates, FTLD-tau, in which tau accumulates, and FTLD-FUS. And as you probably know, frontotemporal dementia is actually a very diverse and heterogeneous disease. So not only in the proteins that accumulate, but also in the symptoms that people may have when they face disease. And this is because, as you can see on the following slides, is because that the frontal and temporal lobes are the regions that are important for different aspects of, including behavior and language, and also motor functions. So it is not surprising that if neurons are dying in these regions, that we can expect problems with one or more of these functions. So this is why individuals with FTD present with personality or behavioral issues, which we call behavioral variant FTD, or language issues, which we call primary aphasia, or can also present with motor disabilities, which include ALS, but also other forms of motor deficits. And if you would put everything on one slide, it would look like this. So on top, you can see different proteins that can accumulate, which, as we discussed, may cause a spectrum of disease symptoms. And maybe you can already see the problem here, which is that there's a lot of overlap, actually, which means that during life, it is very difficult to know which protein is accumulating inside the brain of patients. And on the bottom, you can see that FTD can be either familial or sporadic, and you can already see some genes listed here, meaning that a mutation in the genetic code of that gene would cause disease. And we will go in further detail in a second, but I just want to highlight one flaw of this figure, which is that sporadic does not necessarily mean that it does not have a genetic cause. It merely means that it's a person that has no known familial history of the disease. But this doesn't mean that they can still carry genetic mutation that also occurs in families like progranulin and ST9ORF. So in the next few slides, I will go over how we find these genes in both familial and sporadic individuals. And I would first introduce to you the concept of genetic modifiers. So to identify genes that have mutations and cause disease, we perform genetic studies where we compare healthy individuals with sick individuals, and we look for differences in the genetic code. And one way we can do this is in families where an affected individual is colored in purple, and we compare them to healthy family members. Or we can also do this on a population scale, as you can see on the right side. And what you can see here is that having this purple plus represents the mutation that causes you to be sick. So using this type of genetic studies, we can find causal genes. And in fact, our lab has been performing these studies for FTD for over 15 years, so long before I joined. And it has been a part of the discovery of both progranulin and the C9orf gene mutation. And in fact, many genes that cause familial FTD have been found, which does not mean that it already stops there, as they're also, to better understand, you can move forward, thank you. To better understand the disease, actually, we also look for genetic modifiers or risk factors. And for example, here in these individuals that are depicted with the red arrow, you can see that they also carry the same mutation, but they are not sick. So we can also use genetic studies to look for factors that protect these individuals for disease, which we can then use to figure out why they are not sick. In addition, there is also a lot of variability in individuals in terms of the disease onset or the disease severity, or also the progression of the disease. So some people progress faster, and we can also use genetic studies to compare between these sick individuals. So we compare them against each other to look for factors that affect the disease presentation or the age at onset. So we either have causal genes, which means that a mutation in the gene caused disease directly. And we have genetic modifiers or risk factors, which are additional changes in other genes that have an effect on the risk or clinical presentation of developing disease. So a genetic modifier can both be positive, so it can protect you, or it can also be negative. So it can also increase risk or cause more severe disease. And it is very important to also study these modifiers, as they tell us a lot on which of the functions of the cells are important, and they can also tell us more on why some people get sick at a younger age, and they can also help us in understanding how the disease progresses, or how the disease works, and then a better understanding of how they work may aid in therapy development and drug discovery. And in addition, profiling these genetic modifiers may also provide better genetic counseling for at-risk individuals, which is very important, as it may help to predict whether or not the individual may develop disease. But of course, it does not stop once we have found these genetic factors. So we also need to perform functional studies to understand how genetic mutations will lead to disease and how the genetic modifiers will actually modify your disease risk. And for this purpose, we can use patient-derived material, which may include blood, cerebrospinal fluid, or brain material. And we can also use animal models like a mouse, but also a fruit fly, in which we mimic or make the same genetic change in these animals and then study what happens. And what's actually really cool is that in recent years, there's also an uprising of a new technology, which is called induced pluripotent stem cells, or iPSCs. So here we take skin cells from patients, we reprogram them into cells that are pluripotent. And this just means that we can form any cell type of the body, including neuronal cells, which of course are a very valuable tool to study the disease. So now that I've explained how genetic studies are performed and how we find genetic modifiers, we can move on to the topic of my research studies and also the primary topic of this presentation, which is TMEM as a genetic modifier. And in the TMEM106B gene, there are over 100 changes in the code of the gene at different locations. And as you can see here, all these changes are inherited together. And this means that there are actually two versions of TMEM106B. And each of these versions is called a haplotype. So this means that in the general population, there are two versions of TMEM106B, which we describe as either a risk haplotype, which I will now refer to as like the R in the next following slides, or a protective haplotype, which will be with a P. So TMEM106B was discovered as a genetic modifier for FTD in 2010 in a study from our lab, amongst others, in which we compared the genetic code of over 500 FTD patients. And we screened the genetic code. They included progranulin individuals, and then we compared them with healthy individuals. And in this genetic study, TMEM was found to be associated with FTD with a very strong effect or signal in the progranulin individuals. So what we saw in this study was that TMEM106B, the protective haplotype, was actually more common in healthy individuals, but almost never occurred in the sick individuals with progranulin mutations. And if something is more frequent in healthy individuals, it may therefore be protecting against disease. And this is indeed also what we found in later studies, also in single families. We confirmed that progranulin carriers with at least one TMEM106B protective haplotype have approximately 50% lower odds to develop disease symptoms as compared to non-protective haplotype carriers. And that progranulin carriers with two copies of the TMEM106B protective haplotype almost never develop disease symptoms, which you can also see nicely here on the slide in this progranulin family, that despite carrying a progranulin mutation, these two individuals did not develop disease while they're already in their 70s, and they also happen to carry two protective haplotypes of TMEM106B. Also in the next family, which is actually recently published to raise more awareness of TMEM106B and to really promote genetic testing, you can also see a patient in their 50s with an asymptomatic parent well in their 80s, despite carrying the same progranulin mutation. And of course, the family wished to understand why the parent was not sick. And then the clinicians reached out to us, and then we encouraged them to perform genetic testing for TMM106B. And here we revealed that the parent that carried actually 2 protective alleles of TMM106B, and that while the sibling of the patients also underwent genetic testing and also carries the progranulin mutation, they also have 2 protective alleles, haplotypes, which means that, like the parent, he also may be protected against disease. So we therefore informed this family that the sibling of the patient had reduced odds of developing disease. So as you can see here with this example, is that testing for a genetic modifier like TMM106B, especially in progranulin families, provides better genetic counseling for at-risk individuals. But also, if we understand how having 2 protective haplotypes of TMM106B protects against disease, we may try to mimic this as a therapy. The pitfall here is that progranulin carriers with 2 protective haplotypes do not get sick, and they also normally do not show up in the clinic to participate in research, which is why we need to rely on genetic testing of families to actually gather information about these individuals. Second of all, the second pitfall was that, so you may wonder, if it's so obvious, why isn't this not routinely implemented yet? And this is because TMM has long been an underappreciated protein, because sometimes clinicians do find individuals with a progranulin mutation and 2 protective haplotypes that are sick, so they don't believe it. But they are very rare, so it's not that common. However, we actually do believe that progranulin individuals who get sick despite carrying these 2 protective haplotypes are actually getting sick because of a different reason. And we're doing additional genetic tests on them to see if they have additional factors that may modify their risk of developing disease. And this is obviously also important to know for gene-based therapy, because the disease mechanism in these individuals may be distinct. So for C9orf72 carriers and ALS, there's also an effect of TMM106B haplotypes, but it is not as strong as in progranulin carriers. So C9orf expansions carriers carrying 2 protective haplotypes protects them from frontotemporal dementia, but not from motor neuron disease. And also in ALS, we see that carrying 2 protective haplotypes is associated with preserved cognition in ALS patients. So these results do indicate that there is a specific effect of the protective effect of TMM106B more on cognitive functions rather than on motor functions. So what followed after the discovery was a decade worth of genetic studies. And if we summarize these, actually TMM106B is associated with many neurodegenerative diseases, including Alzheimer's disease, but also Parkinson's disease. And we also established a role of it for in mental health and healthy aging in general. And we, for example, also see that it associates with TDP-43 pathology in a person without FTLD. So we conclude that TMM is a major risk and disease modifier in FTD and other diseases, and that it has a very important role in the aging brain. But this is all on a genetic level. So the question raises, what is TMM, what is it doing, and why is it modifying disease risk? So let's start with looking at what happens when you either have the risk or protective haplotype. So as our current understanding of TMM106B, there are 2 possibilities, which may also happen simultaneously. The first option is that the changes in the DNA causes less RNA, which then causes less protein, or that the changes in the DNA cause an alteration in the protein, leading to a faster degradation and therefore also less protein. So either way, we currently think that the risk haplotype means that you have more TMM106B protein and that you have less TMM106B when you have the protective allele. So if we go back to the figure of the cell, you can see here that the cell has many different compartments with all specialized functions. And studies found that TMM106B is a lysosomal protein, which means that it is localized to the lysosome. And this is the compartment that is responsible for waste clearance and recycling. And the way it does that is by creating an acidic environment in which certain enzyme can function to cut proteins into tiny pieces and then facilitate degradation. So if you can go further, thank you. So TMEM looks like this. So it fits here in the membrane of the lysosome with a portion of the protein outside of the lysosome and a portion within the lysosome. And while you don't need to know the specifics, it's important to already appreciate that it can be cleaved and processed into these smaller portions that you can see on the slide. So when the question raises on how TME106B modulate disease, the fact that TME106B is located in the compartment, important for waste clearance, together with the fact that neurodegenerative disorders, including FTD, are characterized by an accumulation and aggregation of toxic proteins, it may already seem straightforward that TME106B may modify disease by regulating the functions of the lysosome, which regulates the clearance of these accumulating proteins. And indeed, changes in the levels of TME106B causes lysosomal dysfunction. So they cause it that it doesn't work anymore. It results in these swollen and immobile lysosomes that I hope you can see here in the picture. But it also changes the acidity and the correct functioning of the enzymes responsible for cleaving the proteins into smaller pieces. So increased levels of TME106B will cause malfunctioning of the lysosome, and it will reduce its capability of degrading these accumulating protein, which explains why TME106B is important in neurodegenerative diseases. So, but how come TME106B is such a strong modifier in progranulin carriers, way stronger than in other diseases? And this is likely explained by the fact that progranulin is actually also a lysosomal protein, which you can now see on the slide. So progranulin is shuttled to the lysosome, where it is cleaved by enzymes into these functional little granulins, and each have their own function. And individuals with mutations in progranulin have reduced levels of both progranulin and the granulins. And so by regulating the lysosome, TME modulates this process from progranulin to granulins, which means that TME106B actually has a direct effect on progranulin biology. And this may affect the levels of progranulin and granulins even further, which can underlie the strong disease-modifying effect we see in these patients. So for C9orf72, there are two options. So expansions in C9orf causes toxic aggregates, but it also reduces the levels of normal C9orf protein. So TME may modulate disease by affecting waste clearance, or similar to progranulin, but to a lesser extent, C9orf is also important in the lysosomal pathway. So that is also a second option here. So for most diseases, the role of TME106B in the lysosome, which clears out waste, is likely underlying the disease-modifying effect. And for progranulin, this effect is just so strong because of its lysosomal functions. And also for C9orf, this may play a role, but it's not as strong. So recent discoveries also suggest an additional way in which TME may modulate disease. Recent technology, which is called cryo-electron microscopy, allows us to look at aggregating proteins and determine how they look. And earlier this year, scientists tried to look at TDP43 protein aggregates in human brain tissue, but surprisingly, it was not TDP43 that they found, but they found this tiny piece of TME106B that you can see on the slide there, and it aggregates in these forms that you can see below on the right. So it's not only is TME important for clearing out aggregated proteins, it actually also accumulates itself. So we have done some experiments to look at this in the brain, and it looks like the picture you can see here. So you have these dense brown clusters in the brain that is TME106B. And surprisingly, what we also saw is that progranulin carriers also showed a really high abundance of these TME106B aggregates. And considering that progranulin regulates the function of the lysosome, it is actually a double hit to the lysosome, which is likely very important in the disease mechanism. So I have hoped to show you that TME106B is important, and then of course, raises the question whether TMM106B can be targeted as a therapy. But can reductions of TMM106B be beneficial? The problem with this is with this approach is actually that TMM106B are very tightly regulated, and that we found that too little actually causes motor dysfunction. So because it's so important in the cell, it's really important to have the correct amount of this protein. So we really need to know first when TMM106B levels are increased before we can try to decrease them. Which is why we are developing a biomarker assay in the lab to detect these different forms of TMM106B, so we would actually know when to treat patients. We are also using mice and cells from mice to study whether these reductions can be beneficial, and this is actually what I do on a daily basis. So my main project involves a lot of mice, which are different models with different changes in TMM106B. So they differ in the amount of TMM106B they have. They have more, they have less, but they can also have just small changes to TMM106B. And I study these mice during their life, and then we look at their brain health at different ages, and we use them to study neuronal cells. If you can go a little bit, yes. We can also use these mice to look at their behavior. Which is actually really cool. So what we can do, for example, with this thing we call a Morris water maze, we let the mice swim, and they need to find the platform, and they need to learn where it is. So by testing how long they take before they find this platform, we can test for learning in memory. We can also use which we call an elevated plus maze or an open field test. And these are tests in which we can look for anxiety and depression. By timing actually how long mice spend in the closed arms of the maze or like in the corner of this box, because usually mice are very explorative. But when they have anxiety or depression, you will see that they will stay in the corners or stay in these closed arms of the maze. And then we can also test for their motor function by timing the amount of time that they can stay on this moving beam, so kind of like a treadmill. And then we can compare this for healthy mice compared to mice that have changes in TMM106B. So that's actually what I do on a daily basis. So to conclude, genetic studies are very important. I hope I have been able to show you that. We need to identify the primary cause of the disease. But we also need to find genetic modifiers that can affect disease risk or maybe clinical presentation. And if we know what they do, we can try to use them first to study the disease mechanism, but also later maybe we can try to mimic this as a therapy. So TMM106B is very important in the aging brain and in neurodegenerative disorders like frontotemporal dementia. And while I have been able to give this presentation in like a cohesive manner and in just one story, it actually took years of research to uncover this disease mechanism. So I will end with this graph representing a decade's worth of TMM106B research, showing everything I have told you today to really show the progress we have made, but also how long this actually took for us to know. And then I would like to end with some acknowledgements, which include my lab and mostly my promoter, Rosa, as well as all the patients and families that participate in research, and of course also the research support that we receive. And if you have any questions, please feel free to ask now, or you can also always contact me later. I have put on my email and my Twitter. I'm really active on Twitter, so feel free to send me messages. And yeah, you can. That concludes this presentation, and I can answer any questions now.
Wow, well, thank you so much for that wonderful presentation, and to your whole lab. And of course, we all are so grateful to everyone who's provided their samples for these things. There's a few questions in the chat, which I can read out. And I have a few too, but I'll go to some of these first. One's a technical, which I think we can do offline. One is, do you find any age differences in age of onset?
So there have been a lot of replication studies in different sample databases, let's call it like that. So you have the American Association, but you also have an European Association. And then we take these patients and we use them for the genetic studies that I have explained. And there's some variability. And there were initially studies that also showed a difference in disease, the onset of the disease, so the age. But in others, like groups or cohorts of patients, it is not holding up. So while some studies do report it, not all can replicate it.
And I don't know if you also saw that further about sex differences with this protective allele.
I don't know. No, I don't think there is any sex differences here.
Julie, just one thing, there is some literature that says that women are diagnosed with FTD at later, at a more progressed disease state than men, which I think is fascinating from like a social, cultural perspective. They're, like the rules of behavior might live in different places in women's brains, perhaps. So they are acting more appropriate for longer. So they're getting diagnosed later. That's just one possibility.
I think that is also, well, that's not genetically related or anything, or TMEM related, but I think it's also the misdiagnosis of women often, because the problems are taken less serious sometimes, which is still like a gender disbalance, which hopefully will soon disappear. And I think in recent years it's improving greatly, but at least in 20 years ago it was way less straightforward that women, the symptoms that they had were taken as seriously as would have if it was a male patient.
There's another question on any other, so aside from sex, any other demographic variations?
Yes,
such as race? Yeah.
I already said that there is, in the general population, every one of us has either a risk or protective, and you have two, so you either have two protective, two risk, or one of each. And there is some differences in the occurrence of these haplotypes in different ethnicities. So in the Chinese population, actually, there are a lot more protective haplotype carriers, while for just the European population, for example, actually the risk haplotype is the more common haplotype. So it's, there are definitely demographic variations. There's also websites where you can find these. I can always include them too.
I would like to jump in with a question. This thing that the protective mutation is protective for FTD in C9orf72, but it's linked to ALS in C9orf72. There's open questions about penetrance in general in C9orf72.
Yes.
So if the protective for FTD mutation looked very solid for ALS risk, that would be very interesting. So I'm curious about that.
So in C9orf expansion carriers, we found an association with having frontotemporal dementia symptoms, so dementia symptoms, but not for the motor related symptoms. So it did not affect motor neuron disease. It only affected the chances of having also FTD. And then in ALS patients, we actually also saw this. So it does not protect you from motor disease, but it does. It is associated with cognition in these patients. So there is some, the protective effect of TMEM is somehow not really related to motor neuron disease, but rather in the brain, really on cognition.
Totally, very fascinating. I think it would be really cool to see, in these any biobank of C9, what the proportion of the ALS C9 cases had it. Would be very interesting.
It really, in that term, it really depends for genetic studies on which group you take, of course, because if you would look at it like, okay, if you have TMA106B protective haplotype, you will not develop FTD, then you will actually get this enrichment in ALS patients. That is not really true. Just because you do this, always in genetic studies, the thing is that you need to define your group that you take. And depending, of course, on how you select this group in which you perform the genetic studies, this will also affect your results. So this is, also a thing to keep in mind.
Totally. Is this test for the protective or risk halotypes available commercially, or?
No. The family I showed you is actually, I think now, online for a month or 2 months, in which we really, again, try to promote TMA106B genotyping by showing this family and to report them, but it's not routinely implemented yet, and that is really an issue. So every time we go to a conference or give a presentation, we really try and get the message across to test. But I think for now, the way to see or know your TMA106B haplotype would be through genetic testing, either through your research studies, and genetic counselors will also be able to help you with that.
Would they be able to, you think, through a research lab or something, or?
Well, we hope so, and we really hope that they are starting to test. We've put all the information that we need there. And usually what happens is that Rosa is really well known. She has a lot of contacts. So usually what happens is an email in her inbox, and then we advise to test for it, and then we can provide information or how they need to do it or what they need to look at. So.
Yeah, and maybe, can I just briefly comment, because I just want to make it clear, like we usually assist in recommending what needs to be screened, but it's usually a clinical lab. I mean, they are able to do this test. I think this is an important message. It's not being done, but it's a simple sequencing analysis, for example, just like you would do for progranulin or another gene. So they can do this. It's just not part of their routine stuff that they do, but they can. So as long as the physician or the genetic counselor is aware and believes it's important, it can be organized. So I think it's just a matter of talking to the people that you're seeing already for these things and explaining the importance to them. And we are happy to help and explain as well. But in the end, it's better if it's done in a clinical lab, of course, than in a research setting. But yeah, we're we're just trying to spread the word, like Jolien says.
And many of us are in studies with all the great researchers around the world. So when we go in for our visits, I'm sure many of us will certainly start asking, and maybe that'll help too. And then Julie has a question. If you're positive for progranulin, are the chances low that you'll have 2 protective halotypes? Yeah, what is the ratio for carriers? Is it the same as the general public, 20%, or is it less?
Yeah, so you basically, as a progranulin mutation carrier, you have the same odds as everyone else. It does not change. So the only way to really know for sure is to test. But as I said, the protective haplotype is actually the minor haplotype, so it's less frequent also in the general population. But we have seen progranulin carriers that have 2 protective haplotypes, as I've shown you in this family. So yeah, there's no way to know but to test.
And maybe in terms of frequency, I think it's about, let's say, 10 to 15% in Europe and North American populations. The only thing to consider is that often the families that do come to the clinic have already some patients in their family, which suggests that it's not families that are completely where everybody is carrying 2 protective haplotypes, because then they wouldn't have gotten anybody who got sick in the family, right? So I would think that if you have other individuals in the family who have already developed disease, maybe the chances are slightly lower, like 5 to 10%, something like that. But it's definitely a possibility. So it's worth looking into.
Tara, who is a genetic counselor and fabulous, has some questions about naming conventions for these things, but I'm sure we can get that offline. I just had a question about these genetic modifiers that interests me, and just would love to hear your thoughts. So there's families, for instance, C9 especially, where there's like my family, multiple people in multiple generations. And there's also this knowledge of families where, big families where, all of a sudden one person's diagnosed and people are like, where did this come from? And there are these data points of some of these population screens where there's more C9 carriers found than you would expect. That might support lower penetrance. But if what's driving the higher penetrance in these families is these risk mutations, it seems hard to think that multiple people would inherit the same risk factors and the mutation, the causative mutation, generation after generation. It seems unlikely to me, and I'm just curious if there's thoughts about that from people who think about this all the time.
I'm not a geneticist, so I mainly work with the mice and look at little cells and stuff. So Rosa is truly the expert on this, so maybe she can also comment. But it really depends on which factors are driving it, and I'm not sure for C9 or 72 whether there are many of these risk factors actually known or already tested for. But it really depends on the frequency of these common mutations, actually. So if they are very common, it's possible that multiple people will inherit it. It's like ApoE for Alzheimer's disease. It's common, so they can really drive your risk. But maybe Rosa can also comment.
Yeah, I think we don't know yet what other factors may influence C9, but there's been a couple of reports about these double mutations, and we also published this, that I think more often than chance, when you actually start to study patients with C9 or 72, they have other variants, other genetic variants that already were known to carry a little bit of a risk for neurodegeneration. And I mean, it's still complicated because you have family. So, the C9 must be the sort of the primary hit, and it runs in family. So that alone must somehow be enough, you would think. But I think if we start to really study the rest of the genome of the individuals who get the disease, there very often are other things as well. It's just that we don't know all these other things yet. So it's complicated to have the full picture. But my bet is that actually it's kind of modified by other factors, but some of those factors are pretty common, like Jolien says. So then, if they have them in the family, then it sort of inherits it together, and then everybody in the family has the C9, but also has these other smaller risk things, and then together they develop the disease. It may also be that C9 has this loss of function and gain of function, and it's just a bit more complicated. So you have a lot of different things that have to go wrong, and in some people, they might be protected from one of these factors, and then they can just be protected. The other thing is that sometimes the expansion may happen de novo, like may occur from maybe not yet being long enough to really cause disease, and then to the next generation it might suddenly actually become a problem. And in that case, the parents are obviously healthy, and it's a new mutation. And this has also been reported in I think in 1 or 2 families as a suggestion that maybe also it can occur de novo, is what they call it. And this might be an explanation for some of the sporadic individuals, but I think it's more likely that the parents were just protected from factors that we unfortunately do not know yet completely. Yeah.
Very interesting. Thank you for both your thoughts. Wow, thank you so much for this lovely presentation. I'm so happy to hear all the hard work that you guys are up to, and we so appreciate it. Does anyone else have anything before we close off the event? Great. Well, thank you guys so much. And I guess we'll leave it there. Thank you so much, the Rademaker Lab. Very happy to be in touch. Perhaps we can arrange another one on some other topics in the future.
Yes, thank you.